guys, it's Mrs. Smith. We have a really unique lab opportunity. There's been a murder, and we are going to help to solve it. Okay. So here's our victim. Her name is Harmony. She was 17 years old, and last year she was found behind the Roses, which is a popular eatery here in College Hill. I know everyone knows what the Roses is. She was reported missing by her mom, and the next morning she was found by a mother and son who were out taking a walk. Um, the authorities believed that she was murdered by poisoning because the last text that she sent to her mom detailed that she was going out to eat. So just based on her body and some chemicals that they found and all of the clues, they said that this was a murder by poisoning. So based on the cell phone records, the Cincinnati Police Department has narrowed it down to 3 people that they believe could have been with Harmony when she had her last meal. So the first suspect is Mr. Davon Davis. He's known Harmony since childhood. He says that he last spoke with her on Monday night back in September of 2019 to help her with her geometry homework. He says that he enjoyed a quiet meal at home with his family, and he says that he did have La Rosa's, and he said that the meal that he had was a traditional crust pizza with sausage, pepperoni, and bacon. Our suspect number 2 is Mr. Justin Watkins, who's a busboy at Olive Garden. He's claiming that he was at work during the time of the murder. He says he's never met Harmony and that his friend was just using his phone to text Harmony, and so that's why her number is in his phone. He says that he ate alone. And he had bread with olive oil and creamy, buttery pasta. He does, however, live in the College Hill area. He spends a lot of time hanging on Hamilton Avenue with his friends. So Justin is also a strong suspect. Our third suspect is Ladarius Adams. And Mr. Adams says that he does know Harmony, but just casually, like just in passing. He was not with her on the night of the murder, but he was with his friends. He actually was eating at BW3's where he had barbecue wings and celery and Coca-Cola. So they were so kind enough as to provide us with some preserved stomach contents from Harmony. And so I'm going to analyze those with you on camera. And our job is to analyze her stomach contents and find out what her last meal was. And we're going to perform 4 tests to search for the presence of macromolecules. The first test that we are going to perform is called the simple sugar test. In the presence of simple sugars, something called Benedict's solution will turn green or yellow or red. And so what we're going to do is we're going to take the stomach contents, we're going to put some Benedict's solution in with the contents, and we're going to put it in a hot water bath for 3 minutes and look to see if it changed colors. We're also going to test for complex sugars or starches. This would be things like breads, pastas, French fries. In the presence of starches, Lugol's iodine, is going to turn bluish purple or black. So what we're going to do is we're going to put some sample in a test tube, and we're going to add some Lugol's iodine and look to see if it changes colors. We're also going to do a protein test. Proteins are things like meats and eggs. Wouldn't be protein in our diet. And so in the presence of proteins, Burette solution is going to change color to purple. It says pink purple, but in the past, I've always seen it turn purple if there is protein. So what we're going to do is we're going to take our sample, we're going to add some Burette solution and mix it very gently and note any color change. So we're going to do a 4th test, and I actually don't have a slide for the 4th test, but that test is a lipid test, and lipids are fats like oils. And in the lipid test, we're going to take a paper towel, put some sample on the paper towel, and check to see if we see a grease stain on the paper towel. So you'll see a little more about that later. All right, hope you're ready for this analysis, real life biology at work. So what we're going to do is we're going to take some stomach contents. These are the stomach contents of the deceased Harmony, and we are going to test for the presence of several molecules. Ready? Let's go. Remember that there are 4 categories of macromolecules. There are nucleic acids, which we won't be testing for today because you don't get your nucleic acids primarily through food. There are carbohydrates, which we'll be testing for today. There are proteins, which we'll be testing for today. And there are lipids, which we will also be testing for today. Okay, so 4 classes of macromolecules. All right, so let's get started. What I have here are the stomach contents of Harmony. I took those stomach contents and I dissolved them in distilled water so that we can analyze them. Let me just pour these stomach contents into our test tubes. I'm going to try and keep it about the same amount in every test tube. Oops, that's probably too much. All right, so we've got 3 test tubes with our stomach contents. Let me move this around so you guys can see a little better. 3 test tubes with our stomach contents. So let me tell you about the tests that we're going to do today. The first test we're going to do is the Lucas iodine test for starch. I've prepared a positive and negative control. A positive control contains what I'm testing for, and a negative control does not. So in the case of starch, what I did is I took some cornstarch and water, and just some plain water. So I have my positive with the cornstarch and water, and my negative with just plain water. So what happened when I added my iodine? Well, my positive control that had the starch in it turned very dark, almost black, while my negative that did not have starch is this reddish brown color. That's just the color of the iodine. Okay, so you can see the difference between the two. What we're looking for, if Harmony's stomach content contains starch or some other very complex carbohydrate, noodles, breadsticks, we are looking for a very dark color. Okay, I did the same thing in testing for simple sugars. This is called the Benedict solution test. So what I did is I took my sample, I put water in my negative control, I put glucose in my positive control. I want to make sure that it's going to be positive for simple sugar. Okay, simple sugars are like glucose. All right, so what happened is once I added the Benedict solution, I put it in some boiling water, which I have here to my right. And this is what happened. The solution turned orange, and actually it was very red when I took it out. It's been out for a little while. Whereas just the water, which has no sugar in it at all, it stayed blue like the solution. Now, the next test I did was a test for protein. In the test for protein, I used egg, which is a known source of protein, and I used water again for my negative control. So my positive control, my known source of protein, and I apologize, I didn't say the name of this test. It is the Buret test. My known source of protein, when I did the Buret's test, you can see just a tad bit of purple there in the solution. That means that it's positive. My water, which is negative, has no purple in it at all. And finally, I wanted to test for the presence of lipids. I took a paper towel and drew the same size circle, and I added a drop of oil and a drop of water and let it dry. In the case of the oil, it left a stain, left an oil stain there. For the water, you can see that nothing was left there. So my positive control would be the oil, because I knew that I put oil and it's going to have lipids because lipid is oil, right? So when I added my drop of oil, or my lipid, I got a big old stain. Now my water had no oil at all. So because my water had no oil, when I added it to my paper towel, I got nothing. All right. So we are going to perform the tests on our sample of Harmony's stomach contents. I just pipetted it into another container. We'll perform those same tests to see what happens. Let's go. So I've got my test tubes, and they are filled with Harmony stomach contents, and I just need to add some stomach contents to this test tube here. I try to put the same amount of stomach contents in every test tube just to make sure that I got a consistent result, okay? So let's first do the iodine test. Remember, when we do iodine, we are testing for the presence of starch. So on your lab paper, find the spot that says Lugol's iodine test. Okay, this is where we're going to put those results. All right, so I'm going to take my sample and I'm looking for starch, like bread, pasta. Okay, I'm going to add 1 pipette full of Lugol's iodine to my sample, and that's what I got. Let's compare it to my negative and positive control. This is my positive control. Do they look alike? This is my negative control. Do they look alike? So which one would my sample be? Would it be positive for starch? Like, put that down. Would it be positive like these, like the positive sample? Or would it be negative for starch? You decide. Now, next we're going to do our test for simple sugars. And I'm going to kind of turn the camera just a tad so that you can see my boiling water here. All right. So, with my Benedict solution test, I'm going to take my sample. I'm going to add Benedict solution to my sample. I think I'll add a little bit more. Okay. And it goes into my boiling water for about 3 minutes. We'll check on that in a second, okay? Let's move on to our next test. The next one is going to be our Buret test for protein. So I'm going to take a pipette full of my Buret solution, and I'm going to add it to the test tube. And actually, I might do 2. Remember that a positive is purple. I'm going to shake it up. We don't shake like this in a lab. You shake side to side, just gently. Okay. All right. So would this be, here's our sample. Would this be a positive result? Is it purple like my positive result? I'm not looking for yellow. Yellow is because it's an egg. Eggs are yellow in the inside. I'm looking for purple. You see all that purple? Do I have any purple here in this test tube? So would it be positive or would it be negative? Okay, it's not going to be white or clear because it's water. The solution, the contents themselves are this color, like this. But what we're looking for is a change to purple. So did it change to purple or did it stay looking like the stomach contents? Okay, so is it negative, no change, or is it positive? Did it turn a little purple? Okay, there's some purple at the bottom. All righty. We're going to go back to our test tube that's in the boiling water and we're going to see what happened. I'm going to pull it out. Okay, now remember, it's not going to be exactly like my controls, but what I'm looking for is a change. Okay, so would this be positive? Did it turn to orange or green or red? Or would it be negative? Does it just look blue? Okay, so with this is my Benedict's test. So find the Benedict's test part on your lab thing. Find the Benedict's test portion on your lab sheet. And you tell me, is this going to be positive or negative? Did it stay blue? Okay. Now, okay, for my lipid test, that's the last one, I'm going to take something that's round, and actually I'm just going to use this Petri dish like I did before, and I'm going to trace around it. Now, what I'm going to do, and I'm going to put just a little bit of my sample on this paper towel. Okay. And remember what I'm looking for. I just put a little. Remember what kind of stain I'm looking for. So what I'm going to do is I'm going to pause and give this a little time to dry, and then I'll show you what it looks like at the end so that we can compare, okay? I'm actually going to write sample here. All right, so I will be back. All right, so here's my paper towel with my sample. Does it look more like the oil? Uh-oh, adjust. Does it look more like the oil or does it look more like the water? Here's my sample. Remember that the oil would mean that it's positive. Does it look like oil? If it looks like oil, it's positive. This is a test that would be positive. If it does, and if it doesn't look like anything, then that would mean that it's negative. If it didn't leave a mark at all on the paper towel, see the water didn't, it just dried. Did this leave a mark on the paper towel? If so, then it's positive. If not, it's negative. Okay. So in the spot where it says lipids, the lipid test, write down what you see. Okay. All right. I hope you guys enjoyed the lab today. When I see you next in class, we will talk a little bit about our results. Remember that your lab report is going to be due on Sunday at 11:59 p.m. You can come and see me during during office hours if you need any help. I'd be happy to help you. Have a wonderful day. Bye, guys.